Good evening. My name is Jill Traynor. I'm Dean of the College of Natural Sciences and Mathematics here at Sacramento State. I'm pleased to welcome you to this talk tonight. The talk is presented by the Sacramento State Center for STEM Excellence. STEM stands for Science, Technology, Engineering, and Math. The center works with area schools, colleges, universities, and employers to create a seamless STEM pipeline for a scientific and technical workforce. I'd like to extend a special welcome to students from American River College. Welcome to campus. We're glad to see you here today. Do we have any students from Will C. Wood High School this evening? Not this time. Okay, great. Well, welcome everybody. This is the second of our series of public lectures on regenerative medicine. This academic year, Sacramento State will host 4 lectures by prominent scientists in the field of stem cell research. The lecture series is funded by the California Institute for Regenerative Medicine and supports our new master's program in stem cell research. We're very excited this fall to welcome our first cohort of graduate students in this program. Next summer, these students will work with our partner in the program, UC Davis Stem Cell Program, as part of disease teams. The students will work on cures and treatments for disorders such as neurodegenerative disease, spinal cord and brain injury, epilepsy, HIV infection and AIDS, osteoporosis, blood disorders, and burns. We look forward to graduating our first class of stem cell research laboratory personnel in the spring of 2011. It now gives me great pleasure to introduce this evening's speaker, Professor Gerhard Bauer. Professor Bauer is an assistant adjunct professor at UC Davis and director of the Good Manufacturing Practices Laboratory with the UC Davis Stem Cell Program. Professor Bauer was educated at the undergraduate school in Wieselberg, Austria, and at the School of Medicine at the University of Austria. Before coming to the UC Davis Stem Cell Program, he worked in research positions at the Federal Institute of Serum Controls in Vienna, the University of Maryland at Baltimore School of Medicine, the Johns Hopkins University, Children's Hospital Los Angeles, the City of Hope National Medical Center, and Washington University in St. Louis. His research in these various positions has focused on gene therapies for HIV. Professor Bauer is author of 27 journal articles and has contributed to over 8 clinical trials. Please join me in welcoming Professor Bauer.
Thank you very much for this very nice introduction. I really appreciate it. So don't think that I'm stuck up with or anything. Okay, we'll take this out of the question immediately, and we'll have a very nice conversation today about my most beloved research that I have been doing practically since the early 1990s, and I have been in HIV research since 1982, when we actually knew that there was something out there that we couldn't even identify what it was. In 1984, I got my hands on my first HIV test kits. It was not called HIV at that time. It was called HTLV3, as you will remember, because it was wrongly classified into a wrong virus category. And then finally, it was realized that it was a lentivirus. And this lentivirus was finally categorized as the human immunodeficiency virus, which has a lot of counterparts in many other animal viruses. And I want to also tell you that we actually have a species jump of HIV. It used to be a primate virus that actually jumped into the human species. We have HIV-1 and HIV-2. HIV-1 is endemic in Central Africa, was endemic in Central Africa. HIV-2 endemic in West Africa. And finally it got into the human population and was spread all over the world. And this is what we saw in the 1980s. We have made a lot of progress in tracking it down and finding out what it is, but we really have not made a lot of progress in fighting it, eradicating it, getting rid of it. And this is what I want to bring towards you today is, how is there a possibility, will there be a possibility to get rid of HIV? Perhaps. Let's have a look what we have been accomplishing. The facts about HIV. I don't know if you can read all that, but I'll try to read it for you. And if there's anything that I can explain to you, we'll do it after the lecture. HIV is still a major public health problem with more than 33 million people living with HIV worldwide. That's a huge number of people. And I think it's underestimated because there's a lot of underreporting going on with HIV. And once in the body, HIV stays there for the life of the individual, since HIV permanently integrates into the genome of the target cell. The treatment is possible nowadays with highly active antiretroviral therapy called HeartShort. But this is not curative. So what we have today is we have very good treatment strategies. People sometimes call HIV a chronic disease. I refrain from that because if you would not take any medication, you would practically die, as we saw in the 1980s when we did not have any medication. There is absolutely no working vaccine. Although there were some news from a recent vaccine trial that showed a 31% protection from HIV infection in the vaccinated group, and this trial was done in Thailand, and it only recently was made available for us to analyze. And we think that a 31% rate of protection is a little step towards finding a vaccine, but it surely isn't anything that we can utilize currently. Clearly, after hearing that, you really know that new and different strategies need to be researched to attack these problems and develop a curative approach for HIV infection. Gene therapy, something that was very hot in the 1990s, had a little dip in the early 2000s and is slowly coming back. But we stuck to that. And gene therapy is something that is really an approach that's multifaceted. You can do a lot of things with it. So what is gene therapy? It's the manipulation of genetic material, the insertion, deletion, recombination used for therapeutic intervention. The areas of gene therapy are, particularly in the beginning, were severe combined immunodeficiency. Why that? It was an ideal disease to attack. It's a monogenic disease. One gene is lacking. If that's not there, an immune system cannot develop. And guess what? If we can replace that one gene, we can cure that particular person. So this is why this particular disease was chosen to start out with. Lysosomal storage diseases like Gaucher's disease, you can actually make an enzyme that breaks down these stored compartments. Cancer, most gene therapy protocols currently are directed against cancer. You can put genes in that actually repress cancer growth. You can actually put genes in that do something to the cancer itself and shrink it. And that's often a gene therapy vector or a gene that gets directly injected into the cancer. But we're dealing with something completely different. We are dealing with an infectious diseases, and people didn't think in the beginning that that infectious diseases like HIV could actually be attacked by gene therapy. But believe me, HIV is a genetic disease, and I'll show you later on why. So, gene therapy can be applied to such a genetic disease. Many other therapeutic applications are, of course, thinkable and will be coming up in the future. But in most cases, gene therapy means nothing else but the transfer of a gene, which is nothing else but a stretch of DNA, into the nucleus of a cell for therapeutic purposes. For people that are not scientists, what is this DNA? DNA is a genetic blueprint of an organism. And a gene is a small subgroup of this particular blueprint. A gene usually contains information how to make a certain protein. One of many thousands of proteins in an organism, and therefore genes are essential. And every single gene in our organism normally has a certain function to fulfill. And if it's lacking, it's not working right. So, which gene, which cells, whatever should we target for gene therapy approaches? So let's look at the cell compartments. For HIV gene therapy, hematopoietic stem cells make an excellent target. I'm going to explain to you why all this happens. But just keep in mind, currently, stem cells, okay? Stem cells are cells of all kinds, and not only the stem cells that you hear about in the news. So I want to explain to you. What are hematopoietic stem cells? The body needs to produce enormous numbers of new blood cells over its projected life of many, many years. Just think about it. A red blood cell has only about a 120-day lifespan. And we have billions circulating in our blood. So a red blood cell, all the students I'm sure know that a red blood cell doesn't have a nucleus. The nucleus is the factory that makes all the information to make new proteins. So a red blood cell is a disposable little machine. That's all it is. Completely disposable. Lives 120 days and you get rid of it. But for that purpose, you have to make more cells. If you get rid of so many, we have billions of red blood cells circulating. You have to have a mechanism to come up with a replacement for these. So, the body desperately needs a source for new blood cells that cannot be exhausted. And it is not the red cells themselves. They can't divide. They have no nucleus. So, in the 1950s already, and even earlier, the idea of a very unique cell was proposed. Something called a hematopoietic or bone marrow stem cell. Hematos, Greek blood. Poios, making it, okay? So hematopoietic, blood-making stem cell. And now you know where this happens. It happens in the bone marrow. This is a type of cell that, when it divides, can give rise to all types of mature cells, blood cells, red cells, white cells, all of them, just from one cell, and can make more of itself at the same time. That's the unique concept behind that cell. So how are stem cells defined? So here's your stem cell. Okay? And this stem cell can do the following. It can self-renew. This is what this means here. It can self-renew. Okay? But then it makes all these other cells, all these different cell lineages. See, you have suddenly 2 cells here, and they make all these other cells at the end, which end up in mature tissues. And all these cells in between are maturation stages, particular progenitor cells, as we call them. And this holds true for almost all other stem cells that we have in the body. So you have one single cell that can make more of itself. It is multipotential, and it is highly proliferative. So this is what you need to remember. Because we have all types of human stem cells that follow the same principle. Hematopoietic stem cells are found in bone marrow, and here comes the surprise, in umbilical cord blood. Why in umbilical cord blood? It's very interesting what happens to us before we get born. Hematopoiesis, the making of blood, before you are born actually happens in the fetal liver and not in the bone marrow. So the fetal liver produces all these progenitor cells, and these go into the circulation of the little baby. And then finally, after birth, these cells settle down in the bone marrow space because they know where they have to go. They have built-in homing signals. And since these hematopoietic stem cells are in the circulation, we can actually find them in the cord blood of the baby. But don't you think that you harm anything or anybody when you take that cord blood? Because what happens right after the baby is born? What do you cut? The umbilical cord. And then afterwards the placenta gets delivered after the baby has already been taken away and is bathing very nicely and is coming back to all kinds of other places where they have to be looked at because babies need to be examined, of course. Mom is still in the delivery room delivering the placenta. Okay, it's there where we get the hematopoietic stem cells from umbilical cord from. It's normally discarded. But what a source to discard. Goodness gracious, we can't do that. We have to salvage these cells from umbilical cord because they are very, very useful. They are hematopoietic stem cells. And nowadays often it gets collected and frozen for later use. We did not realize in the past what a source this was. But keep in mind, nobody gets harmed when you collect umbilical cord blood. And also, there's often a misconception. Umbilical cord blood has nothing to do with mom's blood. It's the kid's blood. Completely separate and different from the mother. Just separated by a membrane in the placenta. The child is of a different tissue And it would be rejected by the mom if this would not happen, that it's so strictly separated from mom. Okay? Mobilized peripheral blood is when you go to the hospital and have a bone marrow transplant done these days. Normally, you get mobilized. And the abnormal situation is that you have an actual bone marrow aspirate taken. How can we do that? We have agents that allow for these settled down bone marrow cells, stem cells in the bone marrow to be mobilized, lifted off its support and mobilized into the peripheral blood, where you can be hooked up to a collection machine, an apheresis machine, and we can actually collect these mobilized peripheral blood stem cells out. And these are the same cells as you would normally collect from a bone marrow aspirate. Now, all these cells belong to the group of adult type multipotent stem cells. Multi means many, and they can only make the tissue they are designated to make. So hematopoietic stem cells make blood. Make a lot of blood, but they only make blood. Then there's the group called pluripotent stem cells. They are found in the early development of an embryo. They can make all the tissues of the body, but not a complete organism. The only really totipotent cell is after you have actually an egg cell fertilized, and that divides into the morula stage. This is still totipotent, where you can actually make a whole organism. And after that, these cells really don't make the whole organism anymore and are become pluripotent. We have done something very interesting in the last few years. We can recapitulate such a pluripotent cell out of an adult cell. And the thinking behind that is the following. All of our adult cells have all of our DNA that we ever need. It's just shut down to a great degree. And only the cells that are needed in a particular function will upregulate certain genes. And all the other genes will be shut down, but you still have your whole DNA. What if we could upregulate all these early genes and revert back such a skin cell into a pluripotent cell? We did that. So human skin cells can artificially revert it back into the state of early pluripotent cells by the insertion of developmentally early acting factors and signals. And these cells can also make all tissues of the bodies, but not a complete organism. Back to the hematopoietic stem cells. This is how they look. See these fuzzy balls here? These are hematopoietic stem cells. And now you see how they attach to these leaf-like structures here. These leaves, in the past, we called stroma. Stroma is the support in an organ. What I learned in medical school is you have your stroma. This is the organ itself. This is all the fibers and all the cells that sit in the fibers. And then your other cells sit in there. In liver cells, it's the liver cells that actually metabolize everything, but all that is supported by the stroma. We thought the same thing happened in the bone marrow. And when I started out with bone marrow cultures, I took a bone marrow aspirate, put it into a flask, and then suddenly I had a layer of these stroma cells growing on the plastic, and on top of that were these hematopoietic stem cells. We found that these stroma cells are also stem cells. They're called mesenchymal stem cells. And these can make all types of other tissues. They can make cartilage, bone, they can make muscle. And they're also very useful in supporting these particular stem cells. Here, our hematopoietic stem cells. So stem cells on top of stem cells. This is what we find in the bone marrow niche. It's very interesting. But this is our hematopoietic tree. And this is what every lecture sees, and this is what I want you to remember. Here, up, all the way up here is your pluripotent. It's your multipotent stem cell, your hematopoietic stem cell, and suddenly it goes into asymmetric division. It makes more of itself, and that stops right here. But the other cell that's being made goes down into lineage, and see what it makes? All these CFUs, colony-forming units, as we call them. And we make colony-forming units, burst-form unit erythroid. These make all the red cells. These make all the platelets. These make all the macrophages and the granulocytes. And over there, we have all these other blood cells, eosinophil, basophil, tissue, mast cell, until we come all the way over here. And these are our T cells. This is a particularly important part of the immune system. It's our adaptive immune system. This all here is the old type of immune system here that all the old animals also have. These immune cells are not very specific. They can't adapt to the antigen that's coming in really well. But higher animals have developed an adaptive immune system where B cells and T cells actually make antibodies and cellular immune responses to particular antigens and pathogens that come into the body. If we wouldn't have that, we would die from opportunistic infections. Opportunistic because these bugs just grow where they want to grow. And if you can't fend them off, you have a problem. HIV is such a problem because it destroys these T cells right in there. And then you have lost your adaptive cellular immune system. And there's one cell in there called CD4 positive cell, helper cell, where everything revolves around like the middle of a clockwork, where you have one little wheel not working and the whole clock stops. It's exactly like that. You take these cells away, your immune system's gone. Okay, now here's the rationale for stem cell gene therapy for HIV. All of the target cells for HIV, CD4 positive T cells, macrophages and monocytes, dendritic cells, brain microglial cells even, are derived from bone marrow stem cells. If genes conferring resistance to HIV were inserted into bone marrow stem cells, all cells derived from these stem cells would resist HIV infection. How can we do that? It's a nice concept, but you need to put it into reality. Over a period of 20 years, methods were developed to transport genes into the nucleus of cells and insert them into the cellular DNA permanently. Wow, what a feat. When I went to school in the 1960s, people didn't even know what T cells were. Then suddenly I heard about, nucleus, DNA, how does that work? And all the regulation and things like that. In the 1960s, we only had 10 years worth of DNA experience, and we couldn't even do anything with it because we couldn't amplify it. Only with the amplification methods that were PCR, polymerase chain reaction, developed in the 1980s, we were able to develop all the mechanisms needed to get into this direction. So it has only been since the 1980s that we actually know how to amplify DNA and how to make these tools that I'm going to talk about. The most reliable gene delivery vehicle turned out to be a retrovirus. And these are vectors based on such retroviruses. These are RNA viruses, which have been known for a while. But what the unique feature about these viruses is they have RNA that they reverse transcribe into DNA, and then they take that DNA and they permanently insert that DNA that they reverse transcribed out of their own RNA into the nucleus of the target cell. That's an interesting concept. Normally, the body works like that. You have DNA in the nucleus. You transcribe that DNA into RNA. That RNA serves as a messenger RNA. That messenger RNA makes a protein. That protein has a certain function. Now, these viruses work in the reverse way. They come as RNA. They have an enzyme that the body does not have. Well, not quite. I'll take that back. Otherwise, some students are going to hang me up afterwards, okay? Normally, okay, in the big picture, we don't have such an enzyme, okay? These viruses bring that. They reverse transcribe that into DNA. And then they bring another enzyme. And they go with that enzyme into the nucleus and open up the DNA, put their genetic information in and close it up again, and poof, they're gone. But that little stretch of genetic information now makes you a hybrid between virus and yourself in the target cell. It's not dangerous, though, because we have harbored over the billions of years of evolution many, many, many retroviruses, and we have inherent DNA that we harbored from all these retroviruses that has been silenced or is even active and useful because it has been used for other things. So it's a normal evolutionary path, it looks like. Such retroviruses have been around for all of our evolutionary period. So that newly inserted gene then makes the gene product it is designated to make and alters the course of a disease. We harbor such retroviruses now to transfer our gene of interest. And that's the cool thing about it. We just take nature, modify it a little bit, and use it for our purposes. HIV is a retrovirus belonging into the family of lentiviruses. Lenti stands for long. It takes a long time for such viruses to actually develop pathology. Such retroviruses like HIV, such lentiviruses like HIV are very efficient in gene transfer. What we did was we re-engineered HIV in the gene therapy field to become a non-replicating gene therapy vector. HIV-based lentiviral vectors can serve as outstanding gene transfer vehicles, efficiently and safely integrating their payload into human hematopoietic stem cells. Anti-HIV genes were constructed. Once transferred into the nucleus of the target cell, they would make gene products that interfered with the life cycle of HIV in many stages. Hence, we made HIV fight itself, which is an interesting little thing to point out here. Okay, I have only been talking in abstract terms, but I know that you like cartoons, so I made some cartoons. So, this is HIV here. See that? It has a double-stranded RNA in there, and these are the enzymes that it brings. HIV attaches to the cell, and then it dumps its RNA into the cytoplasm of the cell, and this uncoating process is very complicated. Attachment is already complicated, but uncoating is even more complicated. But just keep it in your mind that it works like that, okay? So RNA goes into the cell directly, but it's very unstable. And the quicker it gets into DNA, the better it is for survival. Because RNA gets broken down like immediately. Students that work with RNA know that because their experiments never work. So you have to wear gloves, you have to make sure that you have protection for your RNA in your buffers and things like that, okay? It's very important. I made the mistake myself. Tried to make RNA many, many years ago, and guess what I had at the end? Nothing but fragmented stuff, okay? So learned my lesson. Then reverse transcription takes place as quickly as possible. Actually, the virus brings partially reverse transcribed RNA in. Then RNA is made into DNA very, very quickly. And then nuclear import happens. See, from here, the DNA needs to get into the nucleus, and it has to cross this membrane. And HIV hooks up to a natural transport mechanism in the cell that actually pulls it in. And there you have another enzyme. See that? One's already used up, but there's another one. And there you go. This is now integration. See, that enzyme is the integrase that opens up the DNA and then puts its DNA right in here, closes up, and disappears. And then afterwards, what it does, from this DNA that's integrated, it has all the genetic information that it needs to make new viral proteins, like the capsid protein, and then a long transcript of the RNA is pulled out of the nucleus, and that assembles with this particular enzyme here and that enzyme. Then the particle is finished and it buds out. And then while it buds out, it takes a part of the cell membrane with it and coats itself with it. So it actually disappears as a virus and only looks like a body cell, a very tiny one actually. It's an interesting process. But it gives us many chances with gene therapy to interfere. We can interfere with the binding of the virus to the cell. We can interfere with the uncoding process here. We can interfere with reverse transcription. We can interfere here with the transport. We can interfere with integration. And we can interfere here with all this budding out of the cells. So many, many steps of interference are possible. And some of these steps are unique to the virus and the material it brings. So if we interfere with such steps, we are not interfering with any steps that the cell would normally need to live for its normal metabolism. So here are examples of anti-HIV genes. The first one were antisense RNAs. directed against the HIV messenger RNA. It's the oldest strategy. You make an RNA that's complementary to the HIV RNA. It attaches and does not allow for such RNA to be further processed. RNA decoys, binding regulatory proteins of HIV. For instance, an RRE decoy or a TAR decoy binds to a certain region of HIV, of such proteins, and the function is inhibited sterically because this protein is then inhibited to go to the binding site that it needs to go to. Ribozymes, it's an interesting concept. It's catalytic RNAs. Plants had that. And plants could cut down RNAs using these ribozymes. And they have been engineered for human use. And we can cut down the messenger RNA of HIV. A better approach, however, is short interfering RNAs. It's another innate process that we have that has been conserved for many, many evolutionary ways from the very beginning on. And we can utilize that to very efficiently cut down RNAs that we don't want. Transdominant negative regulatory proteins were used. In one of our clinical trials that we did, we used that. It actually binds up a particular regulatory region on the HIV RNA, and it will not allow it to be transported out of the nucleus. But a very important strategy that I want to point out to you, and I want you to remember, is this. Ribozymes, or siRNAs, directed against the messenger RNA for the secondary receptor for HIV on the cell. And this is a very, very important thing. Ribozymes or siRNA directed against the human messenger RNA for the secondary HIV receptor, CCR5. Why? Here comes. Approximately 1% of Caucasians harbor a natural mutation, a deletion of the CCR5 chemokine receptor. And that's interesting. Why would that be, and just in Caucasians? It has something to do with the Black Plague. This was in the 12th and 13th century in Europe. It spread from Asia into Europe, and there it really got rampant. Rat fleas actually bit people and infected people with Yersinia pestis, which is the bacteria that causes the Black Plague. And people died by the dozens, by the hundreds, by the thousands, by the millions. But a few survived, interestingly, without getting sick. And it looks like that the CCR5 chemokine receptor was involved. When you didn't have it, Yersinia pestis could not grow as efficiently. So these people were selected. And it looks like this is why Caucasians have this mutation. Homozygous individuals with this deletion are also resistant to HIV infection because HIV uses this secondary receptor to actually enter the cell. It needs CD4 as a primary receptor and CCR5 mainly as the secondary receptor. If that's lacking, HIV can't get in. Heterozygotes are also protected to some degree or have less severe HIV disease. And the absence of the CCR5 receptor is not associated with any pathology due to receptor redundancy. And that's the fun thing about it. You wouldn't even know that your CCR5 deleted because there's no pathology associated with it. Now, the knockdown of this receptor makes an ideal strategy for HIV gene therapy. Here goes one more time. HIV particles attack a cell. They attach to it. A virus attachment is mainly mediated by CD4, the primary receptor, and CCR5, the secondary receptor. And here's Dr. Anderson's strategy. He's right here. He developed an siRNA that can almost completely knock down this particular receptor on the cell surface. And he could demonstrate using this siRNA that we completely eliminate HIV infection, just using this particular one gene. He has several publications on that. But that's not all he did. He thought, why not doing a secondary thing? In case HIV is so infectious that it can actually bind and get in no matter what, we can prevent the uncoating of the virus. So if you can't let the RNA out, then there is no RNA to get reverse transcribed into DNA. Guess nothing gets into the nucleus. This would be the ideal strategy Don't let HIV get into the cell to start out with, and don't let it go into the nucleus. Once in the nucleus, it's in there forever. If you can prevent integration of HIV, that's the perfect strategy to prevent HIV from getting in. And in case something gets in, we have to have a way out. And that is inhibition of the transcriptional upregulation of HIV. And we can do that with a tar decoy. Has been used a while and has been developed for a while. But guess what? If we combine all these strategies that Dr. Anderson actually did in one vector, which is a challenge in itself, then we could completely inhibit HIV from replicating at several stages in its life cycle. Like we do with triple combination therapy that's out there and that's working. So the virus can't easily mutate around every single step in that particular therapy. And why can't it mutate? Because you actually prevent it from replication. Why does it not mutate? Well, when it doesn't replicate, reverse transcriptase cannot cause mutations in the virus because it's reverse transcriptase, the enzyme, and its infidelity that makes these mutations possible because it's so dumb that it makes so many errors, that so many new things arise that are different. See that? And if you let HIV run and replicate in the way it does normally, millions and millions of copies, you have many unfit viruses, but then there are a few that are different and are fitter and can get around these agents that you use to suppress it. And that happened with monotherapy, with AZT. Within weeks, HIV gets resistant. Within weeks, when you use a single agent, HIV gets around it. If you shut it down, however, it do not allow it to replicate and make different strategies to get around and mutate around, you have increased your chances to shut down HIV completely by several million-fold. And this is why such therapies work. Okay, at the end, we end up with no particle production, and this could be a really good strategy in a gene therapy vector. And this is what Dr. Anderson did here, okay? This is our normal vector that we use just to report that we get some gene transfer. We have green fluorescent protein in there that's driven by a PGK promoter, and that's our normal vector that we have and that we use for regular lentiviral vectors, okay? In here, he inserted a TRIM5 alpha. That is this particular factor that allows HIV to actually stay in the capsule, prevents it from uncoding. Then we have this siRNA or shRNA, short hairpin RNA, against the CCR5 receptor. And then here's your tar decoy. And now make all that work in one vector. And that's the difficult part of it, because other people's vectors fall apart. It needs a lot of work and modifications, optimization, and a lot of trial to get such a vector to work. We have this HIV-based vector. It goes here into our bone marrow stem cell. Then there is no particle production because these vectors are replication incompetent. It's a hit and run. So you hit the cell, you transfer your gene makes the gene product, which is the anti-HIV genes that it's expressing here. And then you have your transduced cells that make all these immune cells that have all these anti-HIV genes in there. So you have a built-in amplification step. You're practically making a new immune system that is completely resistant to HIV. Is that really working in reality? Absolutely. Look at this. Here's our transduction efficiency. In hematopoietic stem and progenitor cells, we get from 50-60% to up to 99% transduction efficiency. We get this gene in really efficiently with our vector. Here, you can actually see the suppression of HIV growth in our cultures. This is the control, where we don't have working anti-HIV genes in there. And this is with anti-HIV genes. There's no HIV output. And now I have to duck, because you have to see this here. Look over here. We can actually see that there is no integration of HIV in the target cell. There's absolutely nothing here. While all these other cells serve as controls, and you can see that there is integration. We see this over here just as much. So, in short, the triple combination anti-HIV gene suppresses HIV replication and prevents HIV integration. And this is what we hoped for. We don't let HIV get into the nucleus. You can't pass it on through cell division if it's not in the nucleus. See that? So you can actually really have sterilizing immunity. So to put this strategy to the FDA-required test, an in vivo model is needed because you can't just show this in a cell culture. In the past, we didn't have that. There was no HIV model, but in the last 2 years, we actually were able to get an HIV model in a mouse going. This guy's crazy, you'll say. How can you do HIV? It's a human immunodeficiency virus. It's not a mouse virus. Well, we have to humanize the mouse. So a mouse that allows the establishment of a complete human immune system, including functional T cells, was not possible in the past. A mouse model that lets us see individually engrafted human cells, and a mouse model that allows recapitulation of HIV disease and testing of the curative potential of a treatment strategy. And this is what it is. It's the NOTSKID IL-2 gamma receptor knockout mouse, short NSG mouse, okay? That's what it is. And here I'm going to show you what you see. You have CD3 positive cells in a mouse, which is human lymphocytes in a mouse lymph node. Look at how many we get there. Human B cells, human macrophages, spleen T cells. We have a CD4 and a CD8 compartment. And we get spleen B cells, and here's our control. We can establish in this mouse, and we have done this over and over and over again, 80% of human cells circulating in the peripheral blood of the mouse. And these actually have a normal distribution of human T cells. How is this possible, you ask? Are they functional? They are. Because there's a little mouse thymus. And the mouse thymus educates these human cells to recognize the mouse as self. And so we are establishing a human immune system that is now adapted to recognize the mice as self and reject anything else. But this doesn't mean that it's not a human immune system. It is. And we can infect such mice with HIV. It goes a step further. If you want to see individual cells, we have to breed a specific mouse on top of that, which is the NOTSKID MPS7 IL-2 gamma receptor knockout mouse. We take a disease model that's the lack of beta-glucuronidase in that particular mouse, and we breed that onto our NSG mouse, okay? Now, human cells all have beta-glucuronidase, which is an enzyme that detoxifies cells. And if you have this particular disease, it's a lysosomal storage disease. But this enzyme put into that particular individual or animal then detoxifies. So if we put these human cells in, they all express beta-glucuronidase. And the nice thing about it is we can stain for them. And we can see individual cells in the mouse tissue on a completely blank background. So we can actually follow where all these cells go, even into the brain and into the layer that's behind the retina in the eye, which is the regenerative layer. We can see it in a muscle, in the cartilage. We can see it everywhere at single cell accuracy. Do we have any clinical experience with our gene therapy approach? We do. It's nothing new. We worked on it for many, many years, and we have done it with single genes in the past. So we know that we can put such cells into patients, and we know that it's safe. I want to show you a slide here. It's a very old slide from the mid-1990s, where we actually took HIV-infected individuals, took their mobilized peripheral blood stem cells, transduced them in the laboratory with anti-HIV genes, and infected such cells in the laboratory with HIV. And we used 3 different anti-HIV genes, older genes that we have here, over there, and yellow shows you how old this is our control, and it used to be neomycin. We didn't have VGFP at that time, so we used neomycin resistance. We selected such cells in a neomycin cocktail. They are toxic normally to cells that don't have the gene, and then we infected such cells. And you can see in the control gene, HIV infection takes off. And in all of these anti-HIV gene transduced cell cultures, these macrophages developed out of the hematopoietic stem cells resisted HIV infection. This was enough data for the FDA to let us go ahead to do clinical trials. And there you see all of the clinical trials with HIV stem cell gene therapy we did in the 1990s. It was done in adults and it was done in children. We treated the first child in the world in 1996 with HIV stem cell gene therapy in Los Angeles. I want to really ask you to focus on this particular clinical trial. This was the last one that we did. What we were allowed to do at that time was the following. Children actually were HIV infected. Some had a blood transfusion when they were very little, and this particular child did. At that time, HIV medication was already in, and she was on the cocktail. The FDA would not let us take off the children from their HIV medication. So we also were not allowed to do any bone marrow conditioning. All we were allowed to do is take some bone marrow stem cells, transduce them with anti-HIV genes, and put them back into the patient. An experiment we would never do normally because it's not rational. Because you need to make space in the bone marrow to actually engraft such cells. And you have to give HIV a chance to be defeated by allowing such anti-HIV gene-containing cells to have a selective survival advantage in the face of a viral load. What we thought was, we can produce an immune system that can survive a viral load of HIV and finally fight it off. The FDA wouldn't let us, but we did the clinical trial anyway to show safety of our approach. And this is what you see. A child received the transduced cells, and here you see gene marking in the peripheral blood. It goes away after 2 months, just as we expected, and we followed the child out to 11 months, and we thought, okay, that's what we expected, nothing new. What we did not expect was the following. The child did not want to continue the anti-HIV medication because she absolutely hated the taste of it. So she took herself off medication. And this is what happened. Although we put a drop of cells practically into a huge bone marrow space with so many normal cells in there, these anti-HIV gene-containing cells came back. They were permanently integrated into the bone marrow space. When the kid's viral load rose, these are the blue bars here, the anti-HIV gene-containing cells also rose in the peripheral blood, which confirmed our hypothesis that anti-HIV containing immune cells can live and survive and thrive in the face of a viral load. So this is what we really wanted to see. Kid was put on anti-HIV meds again. Of course, viral load dropped. And of course, the gene marking dropped. But this was really something that we wanted to see. So proof of principle of selective survival advantage of anti-HIV gene-containing immune cells could be demonstrated in a pediatric patient who went off heart. But our hypothesis goes even further. If all bone marrow stem cells would express anti-HIV genes, permanent suppression of HIV or a cure of the disease is in all likelihood possible. This is a very interesting experiment that was performed in Germany. The treatment of HIV-1 infection by allogeneic bone marrow transplantation. So what happened was the following. Dr. Hütter in Berlin actually took a patient that was HIV positive who had lymphoma, a blood cancer. Lymphoma can be cured with an allogeneic bone marrow transplantation. You take bone marrow from another person and transplant that into the recipient who has leukemia. You wipe out the leukemia first with chemotherapy, but then, of course, there's no bone marrow left, so you have to give a rescue bone marrow transplantation. And what he did was he took a person as a donor that had the CCR5 deletion. So he managed to find somebody against all odds that was a match and could be used as a donor for that patient. Now the recipient recovered beautifully, is cured of his lymphoma, and for the last 2.5 years had no signs of HIV, no detectable HIV in the blood. So our hypothesis is pretty correct, I think, at least anecdotally confirmed with this one patient, okay? How could we do this with gene therapy? How could we do this in reality with everybody? How can we replace, in a gene therapy approach, all hematopoietic stem cells in the body with HIV-resistant cells by using pluripotent stem cells. How do we make such stem cells? 4 genes. The ones that I mention here and others work too are inserted into the nucleus of a human skin cell via lentiviral vectors. These genes reprogram the skin cells by producing very early acting gene products found in embryonic stem cells. In natural development, such genes, however, get shut down in the long run. And these generated pluripotent cells have very similar properties as embryonic stem cells and can develop into any type of human tissue, including hematopoietic stem cells. Advantage, they allow the generation of a patient's own tissue. So you make your own new hematopoietic stem cells. But now comes our engineering effort. So here we make such iPS cells. Here you have your skin fibroblasts. You actually put these vectors in, you make your pluripotent stem cell, and then you differentiate these into the mature cells. But now our genetic engineering sets in. We take these iPS cells, select one of these, and then we insert anti-HIV genes into such cells. And then such hematopoietic stem cells can be generated and transplanted into the patient, and the whole bone marrow can be replaced with HIV-resistant cells made from that particular patient. You guys are crazy. No, not really. We're working on it. And this is, as far as we are already, we can do this. We can certainly do that. And we can show that we can actually get to that point. Down here we have differentiated hematopoietic stem cells out of these early embryonic stem cells. And how do we make such cells clinical grade? By applying good manufacturing practice. This is me. And this is Brian. Oh no, it's Steve actually. He is very scared because he needs to work with me in the GMP facility. And I tell him, you better do this or nothing's going to come out of your graduate program. So he goes in, he gowns up, he works very hard. He puts all these cells into the incubator. This is actually a photograph of our first gene therapy experiment that we did at Children's Hospital Los Angeles. I keep that slide in there because it was really 20 flasks that I single-handedly put into that incubator and babied for the transduction period. And then all that went into a little syringe and I was given into the patient intravenously. We have now at this, here at our stem cell center a 6 manufacturing lab, class 10,000 GMP facility. Multi-use facility with high flexibility and versatility. Construction started in 2008 and we are now moving in. This is in our CERM Institute for Regenerative Cures at the corner of Stockton and Broadway. And we're ready to move into the GMP facility. See how large this building is here? It's a 100,000 square foot building. And this is where our new students will actually then be educated. Here is our GMP facility. Here are our basic laboratories. This is the large animal facility. This is Dr. Nolter's laboratory, the translational laboratory. And here we'll be able to conduct all the science that our students in the program are going to do. This is the GMP facility, about 6,000 square feet. You can see we have 6 manufacturing rooms here. And we do all our manufacturing of 6 different products at the same time, completely separate from each other. Down here we have a quality control laboratory. We have a shared imaging room here. We can actually image stem cells right in there. And we can actually then pass them safely back into a manufacturing room after we have dissected embryonic stem cells, iPS cell colonies, and things like that. And we have 2 rooms that we can switch in pressurization, where we can actually do infectious manufacturing, important for HIV, and also vector production. And here we have a fax sorter, a clinical grade fax sorter, where we can actually sort out stem cells and other cells by fax. This is the actual work environment. This is a picture from WashU. Soon you'll see a picture of me sitting in there. This is me, and this is Johnny in the background. And here we have our fishbowl window. Actually, we can cart by tourists on the outside so they can look in, see us working there, and at the end is the collection box. Drop your money right here. Now, here are the acknowledgments. I'm within my time limit. I want to acknowledge it goes that far back. Children's Hospital Los Angeles, and all my colleagues, particularly Donald Cohn, my mentor. City of Hope, John Zaya. Very important clinical trials were conducted at City of Hope. And here, this is our disease teams. This is the stem cell program. at UC Davis. Dr. Nolte, Stem Cell Program Director. Dr. Anderson, who contributed to this because he did all the work that I showed here. Brian Fury, who's with us and is working with Dr. Anderson. Steve, who is going to be working with me, also in the GMP facility, making these clinical-grade vectors. John Javian, our medical student. Janine, immunodeficient mouse core. And Amal Campbell, our stem cell core. And all the other people in the laboratory that I can't even mention because there are too many right now. And I wanted to thank you for your attention, and I'll be happy to answer questions. Did I stun you all?
Yeah, CCR5 configurations are
That's exactly what it is. Because it's used as a chemokine receptor. Chemokines are inflammatory cytokines practically that are released when you have an immune reaction or an injury. And many other receptors can take over. We have so many other chemokine receptors that it looks like they're triply, quadruply present. Because it looks like it's very conserved that you have to have these chemokine receptors. And if one is lacking, there's no loss in function. Absolutely none. You wouldn't know. Maybe you are deleted. You don't even know. Yes?
do a lot of biology engineering in my career. Don't primates already have that, or primates?
So, primates are number one, very difficult to work with. Number two, they have a much longer lifespan. Number three, I think it's unethical to use primates if you don't even know if your experiment's going to work. Okay? So what you want is, you want to have an easy system with relatively high numbers of individual animals that you can use, so you have statistical significance. What you also want to do is, you want to make it repeatable. You also want to make sure that you have a system that you can apply close to your laboratory and don't have to drag out your cells to another facility all the time. So if you have a mouse, it makes life much easier. And most people don't have a real problem in using mice compared to primates. Yes?
Did you introduce mice yourselves, or was that previously?
No. So Jackson Labs did. Jackson Labs is the mouse supplier normally, and it was not engineered. It was a fun coincidence that one of these mice had a deletion. And then they figured out, oh my God, they don't have any functional T cells anymore. Now we have everything we want, that we can engraft human cells without interference. And this is what happened. And this is a very lucky coincidence. But we did cross the MPS 7 mouse onto the IL-2 mouse. We did do that. Yes?
Is there any concern that the new retrovirus that you created itself could undergo a mutation and somehow become virulent to the host?
It's a very good question. We are always worried about everything, and so is the FDA. Okay? So, what we do is a lot of safety testing. Dr. Anderson spent hours and hours and hours of showing that actually there is no other viruses emerging, there's no other things, there's no replication competent virus. If they are not replication competent, they can't make more of themselves, see? Then these viruses are also safety modified. What I haven't told you is that we spent the last 15 years practically modifying lentiviral vectors so that they are in the 4th generation, I think, right now, and actually are very safe. And we can use them without having to fear that recombination occurs. We also have a split plasmid system for making our retroviruses, which is actually putting the structural proteins on one plasmid, putting the envelope on another plasmid, and putting also the inserted gene on another plasmid. And they are going to be in different parts. So the recombination of all these genes, when they are in different places, is very difficult. And then we also have self-inactivating lentiviruses, which actually in reverse transcription, it moves its LTR over on the other side, and the LTR that's moved over is inactive. So it's a lot of engineering that went into such vectors. It's a bit complicated, though. And sometimes when we bring this up in lab meeting, Jan says, oh, go and talk to Joe about this. I don't want to hear about it. Yes?
Where do you see moving into clinical trials with people?
For the regular hematopoietic stem cell gene therapy approach is happening currently. So we can actually take hematopoietic stem cells from patients. We are negotiating with our bone marrow transplant doc currently, and we want to actually use the GMP facility to produce our new anti-HIV vector clinical grade, and then do the same approach that we had in the past. You saw our transduction efficiency. It's pretty efficient. And if we can do some marrow conditioning and make space in the marrow, we may be able to actually really get a clinical benefit going, particularly in patients that can't take medication. Particularly in patients that are allergic to HIV medication, that have no other way out. That would be the group to target, and this actually happens. It's an interestingly high number of patients that cannot take medication. Yes?
The question first, though, do you know specifically where in the human genome that the anti-HIV genes are inserted?
So any lentivirus has a quasi-random integration pattern. So what happens is HIV just doesn't know where it integrates. It integrates where it easily can, but it has preferred spaces where it integrates. The good thing is these spaces are normally not in any place where it interferes with the function of a cell. And we have a natural experiment going on. We have 33 million people infected with HIV. And in these 33 million people, we haven't seen HIV causing directly any cancer. Yeah. So it's very interesting. HIV and its quasi-random integration does not seem to cause any genomic disruption that causes cancer, which is not true for Maloney leukemia virus because it preferentially integrates into active start regions of genes, which HIV doesn't do. And so it does not get continuously upregulated, which is very interesting. So HIV seems to be a much safer gene therapy vector with quasi-random integration. I don't hear you.
There's an article that came out a couple months ago from the Children's Hospital, and apparently the placenta is also in addition to cord blood is also the source of why.
It's the same thing practically. Yeah, you can get it out of the placenta, you can get it out of the cord blood. I was not able, of course, in this short lecture to give you all the sources of such stem cells, but there's many, many sources of stem cells that you can actually tap, and placenta is, of course, one of them. Yes?
How long does the process take from the time that, say, you draw a sample of bone marrow to injecting the new genes, to grow the culture, to put it back in the patient? How long from start to finish does that process take?
Joe has it down in less than 24 hours. Yep. That's what we want. The shortest culture period possible because you don't do any harm to the cells in culture. But you have to have them out for a little bit. We can hit them with a relatively high multiplicity of infection, get enough in with efficient vectors that don't have any dead particles in there, and then put them back into the patient. And the final step of actually getting it into the genome of the cell is happening in the patient in its natural environment. Done? Cool. Thank you.